Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about vaccines. I previously talked about whole pathogen vaccines. Now we're on subunit vaccines. The subunit vaccines contain these five subcategories: polysaccharides, toxoids, recombinant protein, viral-like particles, and nanoparticles. So we'll discuss each one of these. Subunit vaccines contain components or antigens. So it's not like in the previous discussion where it's the entire bacteria or the entire virus. It is going to be a portion of the bacteria or a portion of the virus. Since the whole pathogen is excluded, this is considered a safer option, but it often requires adjuvants to help elicit a stronger protective immune response. The original pertussis vaccine in the 1940s consisted of an inactive bacterium. Pertussis vaccine helped fight against the whooping cough. It caused minor issues like fever and swelling at the site of the injection. So people did not like this inactive whole bacteria. In the 1970s, many people avoided the vaccine because of the minor issues. Then the NIAID developed a non-bacterial vaccine. This one did not have the bacteria. It was acellular, and it was a pertussis vaccine. So this component vaccine had fewer side effects. This is the bacteria that causes pertussis. So in the beginning, we used an inactivated bacteria. It caused several side effects. The use of the vaccine declined. People did not want to get the vaccine. So then we created these subunits, these components of the bacteria, and it successfully worked. So bacterial vaccines are made of components such as sugars that might make up their cell wall, polysaccharides, secreted proteins, and even toxins. One example that we'll discuss is the polysaccharide vaccines. An example of a polysaccharide type is Haemophilus influenza type B. They commonly call it HIB vaccine. This is a polysaccharide vaccine, a subunit of this organism. So Hib is one example of a polysaccharide vaccine. Here are some other pure polysaccharide vaccines. There's one for pneumococcal disease, there's one from meningococcal disease, and there's one for Salmonella Typhi. Immune response to polysaccharide vaccines are independent of T cells. So the B cells are stimulated without the T helper. Because you do not activate the T helper cells, this is going to cause a lower titer of antibodies to be observed when using the polysaccharide vaccines. Boosters of polysaccharide vaccines do not increase the antibody production. It does not produce the amount of antibodies seen. Polysaccharide antibodies are less efficient. Most immunoglobulin M production is observed with polysaccharide vaccines, and these are the short-lived immunoglobulins. And we only have very little immunoglobulin G that will be seen when using these polysaccharide vaccines. And the IgG is the long-term immunoglobulin or antibody. In the late 1980s, the solution to low antibody titer was discovered in reference to the polysaccharide vaccine. They discovered that if you join the polysaccharide vaccine to a protein, it increased the antibody production. So a conjugate was formed. This protein conjugate initiated a T cell dependent immune response. And when you activate the T cells, especially the T helper, you therefore will increase the amount of antibodies that will be produced. The first conjugated polysaccharide vaccine was for the Haemophilus influenza, Hib, and this was in 2000. Unfortunately, Hib is no longer used in the United States. We also have meningococcal conjugated vaccine that was licensed in 2005. The second type that we will discuss is secreted toxins. So secreted toxin types are for diphtheria, pertussis, and tetanus. The antigenic component of the molecule are chemically inactivated. It is called a toxoid vaccine. So I have two pictures of the diphtheria toxin and the tetanus toxin, and it is going to be chemically inactivated, chemically modified. This is going to be the toxoid vaccine. This toxoid vaccine stimulates antibody-mediated immunity, but it requires multiple doses because they possess few antigenic determinants. To increase the immune response, the toxoid is mixed with adjuvants. These are binding agents such as aluminum or calcium salts. These adjuvants help to precipitate the antigen and hold it in the tissue, therefore releasing it gradually into the bloodstream. The next type of subunit we'll talk about is recombinant protein vaccines. And we have 5 types that we'll discuss. The first 3 are hepatitis B, human papillomavirus, and influenza. So 3 of these recombinant protein vaccines are created by inserting viral genes into a yeast cell. Therefore, the yeast will produce the protein that your immune system must see and identify as foreign to build the immune response against. The 4th genetically recombined vaccine is for Salmonella Typhi, which is a bacterium. And it has been genetically modified to be avirulent. So the live typhoid vaccine is TY21A. This is a recombinant vaccine. The 5th recombinant vaccine is from a live attenuated influenza virus. This is used as a nasal pharynx application. The fifth type of subunit we will talk about, subunit vaccine, is virus-like particles. So proteins from the outer shell of the human papillomavirus can elicit an immune response, like the natural virus from the 1990s. So the protein is called the virus-like particle. The sixth one we'll talk about is nanoparticles. So ferritin is an intracellular blood protein that stores iron. This protein can self-assemble into nanoparticles. These nanoparticles can display a protein antigen. So these are all the different types of subunit vaccines that can be used. These are some common examples of such subunit vaccines.